Okay. Kakru?
Yes, ma'am, you can start now. You can start. You can start, ma'am. Hello. Shama, ma'am, you can start now. Many students are still joining, so wait for 2 minutes.
Okay, sir. Okay.
Yeah. 45 students have joined up to the time. And sir, it is not audible. One has written. It is audible. The problem may be with your device. You check it, problem is with your device. I think Shama ma'am has not joined. I request everyone to mute your mic. Yes, more than 50. So very good. More than 55 have joined. More than 55 have joined. Now, Shama ma'am, you can start. Shama ma'am.
Yes, sir. Yes, sir. Good
Start
evening,
now.
everyone. Good evening, everyone. I would like to welcome Dr. Anil, sir, from Hyderabad, and Yogita Lokhande, ma'am, for a brief introduction of Anil, sir. Yogita, ma'am?
Yes, ma'am.
Assistant Professor Microbiology, for a brief introduction of Anil, sir.
Thank you, ma'am.
Welcome.
Good evening and warm welcome to all. Hello.
Yes.
Okay,
yes.
okay.
And warm welcome to
Everyone
all.
else, mute your mic, please. Yes, Yogita, start.
Good evening and warm welcome to all. Myself, Yogita Lokhande, Assistant Professor, Department of Microbiology of Swami Sri Swarupanand Saraswati Mahavidyalaya. Today, we are here for the lecture on how to prepare for entry in biotech industry, organized in joint ages of Swami Sri Swarupanand Mahavidyalaya and Microbiology Society, India. It is a matter of honor for me to introduce our eminent speaker of the day, Dr. Anil S. Yadav, sir. Sir has completed his Bachelor of Science and Master of Science in Chemistry and Doctorate in Philosophy in Chemistry from OPJS University, Rajasthan in the year in 2019 and have 29 years of working experience. Sir is currently working as Associate Vice President at Biological E Limited, Hyderabad, where he leading and managing a team of functional heads and established the anti-sera division. Sir has expertise in the area of anti-snake and scorpion venom, COVID anti-serum, gas gangrene, and many more. And he also published 2 research papers in peer-reviewed journals. With this brief introduction, I hereby request Dr. Anil S. Yadav, sir, for his lecture. Please, sir.
Good evening, everybody.
Yes, sir, you are audible.
Can I audible?
Yes, sir, you are audible.
Good evening, everybody.
Good evening, sir.
Thanks for given opportunity to Microbiological Association of India, and thanks to Dr. Deshmukh sir. He is my guru. He is my professor. I was completed my graduation with under Dr. Deshmukh sir, 1994, and now can we start that?
Yes, sir.
Okay. Actually, today I will.
Deshmukh sir, we can start now.
Yes, yes, yes.
Okay.
It's good,
Okay.
good going, good going.
Yes, sir.
Actually, last 29 years, we are going to take the interviews and pressures, experience, and all these things because we are leading the team. We always involve in interview of all senior, juniors, and middle management people. So based on that, what we did, all biotech and biopharmaceutical and pharmaceutical companies, maximum pressures required now. So that we generalize, we asking the questions like HR, admin, and technical team. Suppose somebody want to join the QC, then QC team lead is there for interview. If they want to join QA, then QA lead team lead is there for interview. We along with HR admin and one more guy, that safety also now that started. If want to go for production area, same thing, production lead and along with all safety team. So always 4 to 5 people is in that interview panel. Based on that, primary they are asking questions as per my knowledge and data collection from all companies. What they are, because our seniors are there and juniors are there, who is asking the what type of questions and how that questions give the answer for pressures. So this is the presentation is there because all pressures are facing the problem before going to interview. How they know to they should know the how to answer the questions and what type of interview is there. So if they are going this question and answers, they will definitely select with their background, their intelligence, and their hard work. So I'm starting this questions. Introductory question. HR is asking the questions. Tell me something about yourself. How did you hear about these positions? Why do you want to work here? Why did you decide to apply for this position? What is your greatest strength? What are your strengths and weaknesses? What do you know about this company organization? Why should you hire? Why should we hire you? Another introductory questions. What is your greatest accomplishment? What are your salary requirements? What do you any questions for us? What are you looking for from a new position? Are you considering other position in other companies? What is the professional achievement you are most proud of? What kind of working environment do you work best in? Where do you see yourself in 5 years? So this is the general introductory questions. After technical questions, I will share that answer also. So this is the technical questions. Explain the part of compound light microscope used in the pharmaceutical microbiology laboratories. Answer is illuminator light source of the base of the microscope enable better view of objects. Condensers, this is the two lens system that collect and concentrate light from the illuminator and direct it into the iris diaphragm. Because these questions used in microbiology, microscope is using in microbiology area. So they are asking this question. Iris diaphragm regulate the amount of light enter in the lens system. Generally, what happens? Student knows that function and all these things, but technically they know only how to work with microscope, but they don't know technically what are the questions this required. If these questions they knows, then they understand this guy is highly technical and addressing the issue after during work. If anything malfunctioning, this question answer is required for explain to the vendor who is supplier the microscope. To the stage, this is the platform for the slide with hole in the center to let light from the illuminator pass through. It consists to clips the hold slide less. Eyepiece to view objects to magnify the object as per the rating on it. Another one, what are the important nutrients of culture media that affects the cultivation of microbiology? In all pharmaceutical companies, biopharmaceutical and biotech companies require the nutrition media for growing the microorganism, for sterility testing, media simulation, and plate count for area monitoring. The answer is the key nutrition of culture media that affects the cultivation of microorganism and source of now carbon dioxide, nitrogen, water, inorganic phosphates, and vitamin and trace materials. Another question, whose name recognize the father of culture media? Generally, all microbiologists knows these things, but they during interview they are missing this because who is taking interview, they are working with this media and culture. That's why they are asking the simple question. Robert Koch is the answer. Discovered the base fresh beef serum, meat extraction bouillon. The term broth of liquid culture being analogous to broth soup procedures and growth. Another question, who was the first scientist cultivate the microorganism on growth media? The French chemist and microbiologist Louis is the answer. Which factor of culture media affect the cultivation of microorganism? 1 minute or so. Madam, can you adjust this video, our photos video? Because screen is not, half screen is visible.
Okay, where is that video?
Not video, it's our photos, right side.
Okay.
1 minute, hold.
Okay. Okay, sir. Okay. Now?
Sorry for this. 1 minute. Now, clear नहीं आ रहा है.
Now? Now, sir, is it clear?
It's clear. Okay. Who is the first? Sorry for disturb. Hello, now clear?
Yes, sir.
Okay. Who is the first scientist cultivated the microorganism on growth media? The French chemist and microbiologist Louis Pasteur in 1992-1985. Which factor of culture media affect the cultivation of microorganism? Culture media factors affecting the cultivation of microorganism are optimum nutrient, oxygen, or other gases, moisture, pH, and temperature. Another question is, Petri dish is named based on which scientist contribution? Julius Richard Petri, 1852-1921. Another question, explain endpoint method of growth promotion test for broth media. In this method, very low level of inoculum are added to multiple tubes of the two media under testing. Growth frequency is compared between the two media to understand the equivalency. Next question, explain most probable number method of growth promotion test for broth media. This is a microbial limit test. In this method, the unknown sample is prepared in a 10-fold dilution series and added to nutrient broth in replicate tubes. The tubes will then either turn turbid growth or remain clear and allow for an estimate of the most probable number of microorganisms. So this test is very important in microbiology, releasing the batches, release the product, and environmental monitor. Why this test is required in manufacturing company for growth promotion test? Because media is working or not, that we want to test in our area before going to use in batch release, yeah, sterility testing, yeah, plate exposing. Another question, explain the kinetic parameters method of growth promotion test for broth media. Answer is, the growth promotion test of two lots of broth can be compared by measuring the growth curve of the same inoculum growth side by side. The growth rate of an organism can be determined by the spectrophotometrically or by viable count. Next, what are the different types of culture media in pharmaceutical and what are the purpose of those type? Nutrition agar or broth and trypton soya agar or broth. Trypton soya agar equivalent to soybean casein digest media. Uses environmental monitoring, isolation of cultivation of non-fastidious and fastidious microorganism. Note, fastidious bacteria are bacteria that need special nutritional supplements and conditions to growth on agar plates. Non-fastidious bacteria are bacteria that do not need special nutrition supplements and condition to grow on agar plates. Trypton soya broth, sterility test. Trypton soya broth, what is the use of trypton soya broth? That is used for sterility test as a general growth broth in microbial enumeration test as well as used for media simulation trials. Why vegetable peptone broth media filling trials? Here media filling trials means before going to manufacture any product, validate the process. All pharma and biotech industries to do the media fill trials as per their process, how they are going to fill the product. So there we want to go for media fill. We are making the media at 121 degrees centigrade, 3% media, and do the autoclave before going to filtration. Then we are going to filter the media by 0.2 micron filter, because that sterile 0.2 micron filter is giving SL. Sterility assurance level is high after filtering 0.2 micron media. Then we are going to start the filling in actual media in vials. That is the minimum size of fill volume, middle size of fill volume, and maximum size of fill volume. That 3 runs we are taking of each size of vials, and This media, if pass all sterility aspects and growth promotion test, then only that filling and manufacturing area is validated for future production. Same thing for fluid thioglycolate media used for growth of bacteria, aerobic and anaerobic as a part of the sterility test. Suboard dextrose agar or malt extras agar using for fungi test. Then what are the different types of culture media used in pharmaceutical and what is the purpose of those? Type R2A. My use is microbial examination of water. This is a low nutrient agar used for the cultivation of heterotrophic microorganism. Another type, Columbia blood agar, detection of hemolytic reaction by staphylococci. Few questions on growth promotion test. What is the promotion of growth? Promotion test of microbiology, culture media. What should be the acceptance criteria? Answer, media to be inoculate with microorganism less than 100 colony performing units. In short, that is called as CFU. Use not less than 5 units strain as recommended by the pharmacopoeia as Indian pharmacopoeia, BP pharmacopoeia, and two organisms from environment isolates or rotation basis. It should be within the 5 passages from the original reference culture seed plots. Compare the growth on the media with media previously approved culture lot. Acceptance is no more than the factor of 2 difference in productivity ratio calculations. Few questions on growth promotion test. What is the calculation of productive ratio and what is the acceptance criteria of agar media plate or solid media for growth promotion test? Productivity ratio, mean of 2 test plate CFU divided by mean of 2 comparative control plates, and acceptance productivity ratio should be 0.5 to 2, that is the equivalent to 50 to 200% recovery. What are the quantitative techniques of growth promotion test for solid media? Quantitative techniques of growth promotion test are of 2 types, econometric, Miles, Mishra, drop count. What are the quantitative techniques of growth promotion test for broth media? Answer is quantitative technique of growth promotion test for broth media are of 4 types. Copious growth, endpoint methods, most probable number, and kinetic parameters. What are the steps involved in the manufacturing culture media? 1. Collection of precocytes, decide batch size, vessels for holding or dispensing the media, weighing balance, petri dishes containers, dehydrated media. Then another one is rehydrator of media. Media powder is rehydrated by mixing the media in the volume of water. Amount should be based on manufacturer instructions. Media manufacturer instruction. And that water is we are using for mixing media is most important. That should not be less than 80-degree temperature. Homogenize the solution by mixing. Care must be taken to avoid the scorching the media. The media should clarify near boiling 95 to 100 degrees centigrade, and the media should only be allowed to boil less than 1 minute. Another question, what are the steps involved in the manufacturing of culture media? sterilization, addition of supplements, depending on the type of medium requirement, filling in containers, status labeling. Few example of good practices in microbiology laboratories. Here I want to say something apart from this. By birth to end up your life, we are using the good practices in our home also. I'm giving one example. After we are coming from our office, yeah, college, school, before going to reach in our home, we are first removing the chappals, shoes, or slippers in out of our door. That is the first GMP. That is called as good manufacturing practices. After that, we are entering into our hall, yeah, in common room. There we are removing our outside clothes, what we use the whole day. Then we are going to washroom. We are clean our hands and foods, and then we are wearing our inside home inside clothes like nighty and t-shirts and all these things. After that, we are going to kitchen, yeah, take the tea, yeah, dinner, lunch, and whatever. This is the same practices, but by written processes, we are using this in manufacturing area. Like when we are enter into the manufacturing company on the gate, first we are going to punch your card. After that, we are going enter into the first where you are working, like QC, production, QA there. Then you are wearing the street clothes, removing the street clothes and wearing company clothes. Why this required? Because whatever contaminated throughout your surrounding area before reaching to company, that contaminated particles and all organisms, bacterial infection, that you should be keep away from your working area, because maximum we are manufacturing life-saving drugs and injectable products. So that should not be increase the load in your manufacturing area. That's why we are removing street clothes and we are going to clean area by wearing the company clothes. Then after wearing the company clothes, there again classification is there. For classification, I'm requesting to all students, please go with WHO GMP guideline. They are clearly mentioned there. What are the classification, what they are required, and they are given in WHO GMP classified area from start to end. What is the positive classification, positive air pressure? What is the negative air pressure? What are the count is required? Which count in which area there's control limits is there. So this basic WHO GMP guideline, if you read for injectables, if you want to go for injectables, if you want to go for tablet and capsule manufacturing, there also they have given. This is the basic idea. If you are getting before going to interview, if you read WHO GMP or Schedule M, that is also good for understanding before going to interview. Then next is frequently dispensing sterilization equipment and media should be maintained and stored in such a way that it should not be contaminated. Frequently disinfectant hands and working surface while carry out the aseptic operation. What is mean by aseptic operation? Aseptic operation means before going to sterile filter in that area, there all equipment, your your hands with gloves, your dresses, and your container and your equipment should be maintain the sterile condition. That is called as aseptic operation. Another thing is fly, rodent, and pest control is most required. Use personal protective equipment such as laboratory coats, safety glasses, and gloves. This is the requirement for self-safety and product safety and environment safety. Drinkable and eatable should not be allowed in the laboratory in the manufacturing area. Sterilize the contaminated used media and waste prior to disposal. Pipetting should not be done mouth. Now, earlier, 10-15 years back, maximum manufacturer and QC chemists are using mouth pipette, glass pipette, but now that trend and automatic micro pipette is available in market. Everybody is using is a high accuracy and high safety. Why aseptic technique should follow in microbiological laboratory to prevent contamination and cross-contamination in microbiology laboratory. Probability of contamination in the laboratory is the sample, media, environment, and facility, etc. How a species can be achieved in the laboratory? Frequent washing and disinfectant hands. Our hands is highly contaminated. That's why always washing and disinfectant the hands. Use of unidirectional airflow station for critical operations, that is called as LAFU, LAFU, laminar airflow equipment. Hand of positive control at the end of analysis. Handling of positive control at the end of analysis. Not touching the samples and accessories directly by hand. Tanya. What is pure culture? A pure culture in a population of the cells or multicellular organism growing in the absence of other species or types in which cells are genetic colonies of one another. What is the most common method to isolate individual cells and produce a pure culture? The most common method to isolate individual cells and produce a pure culture is it to prepare a strict plate method. Explain the strict plate method is another question in microbiology. Sterilize the inoculating loop by heating it until red hot in a flame. Until it is red hot, allow it to cool. Pick up a loop full of liquid inoculum or bacterial growth from the surface of an agar plate and starting about 2.5 centimeter in from the edge of the plate, streak light back and forth with the loop flat, making flows parallel, streak back of the edge of the plate to quarter of the plate. Repeat the same process for remaining 2/4 of the plate. Flame and cool the loop again. So this is the picture how to do that straight plate method. What are the different staining techniques used in the pharmaceutical microbiology laboratory? Gram staining, bacterial spore stain, fungal staining, Gill-Neslin stain. This common question, current new freshers, now if we are asking the question, they are not telling one technique also name. So that's why these 4 techniques they should tell the after asking the question. What is the first step of microbial identification? The first step of most identification program is colony and cellular morphology of the microorganism. Colony morphology is the method that used to describe the characteristic of an individual colony of bacteria on agar media in the Petri dish. Colony morphology is normally classified based on the form, elevation, and margin. So some of photos is there. Explain the principle of the gram straining technique. 70 to 80% pressures not giving this answer correctly. So that's why we consider this question is very simple, but because of their knowledge and all these things, they are not giving clearing. So this is the clear answer. Step 1, crystal violet primary stain aqueous solution consists of positive and negative ions. These ions penetrate through the cell wall and cell membrane of all type of bacteria. The positive ion stains the bacterial cells and stain the cell purple. Second, iodide, the moderate, interact with crystal violet and forms complexes. Step 3, decolorization made of acetone and alcohol 95%, interact with the lipid of the cell membrane, a gram-negative cell losses, its outer liposaccharide membrane and the inner peptide glycol layer is left exposed. On the other hand, gram-positive cell becomes dehydrated because of property of decolorize. The complexes trapped within the gram-positive cell due to the multi-layered nature of the peptide glycan, peptidyoglycan. Step 4. When decolorization is added, the gram-positive cell remains purple and the gram-negative cells loses its purple color, loses its purple color. Step 5. Sapronine, the counter strain is used to provide color to gram-negative bacteria, a pink or red color. Explain the principle of the gram straining techniques. This is the sum of photo. What are the common source of pyrogen? Non-bacterial source and bacterial source. Non-bacterial source, antigen, polynucleotide, steroids, adjuvants, viruses, fungi. Bacterial side, bacterial source, streptococcal toxin, staphylococcus enterotoxins, mycobacterial cell wall components, bacterial cell wall, liposaccharides, that is liposaccharides is giving endotoxin. So this is the technical questions for pressures. Then again, I'm coming back to these interview questions. For HR is asking common questions. What first 2 slides were given, tell about yourself. First question from HR, yeah, any interviewer. This is one of the questions that most people hate, at which they could avoid, but on the most part, the majority of interviews.
Hello, Dr. Anil. Hello.
Yes, sir.
Dr. Anil, I have doubt. Hello, Dr. Anil.
Yes, please. Hello. Hello.
Sir, I have small doubt, sir.
Please. Hello.
You can ask.
Am I audible, sir? Hello.
Yes, please.
I'll just ask the question later, sir. I forgot.
Hello.
Okay, he will ask later on, sir. He has forgotten the question.
No,
You please
I'm
write in the
noting
chat
down the
box.
question, sir. I'll ask at a time, sir.
You
At the last.
please write down in the chat box.
Yes, no problem. I will share that your answer by mail also. Hello.
Oh, okay, sir. Thank you.
Okay.
So
Then they mention a little bit about your past, relevant to the skills and experience.
Sir,
Hello.
Continue, sir.
Can I audible? Madam? Can I audible?
Yes, sir, you are audible.
Okay. What are your weaknesses? Here, many people, they are thinking, what should I do? What should I think? What should I tell that? What are the weaknesses? You should be here clearly by your uh, own judgment and own your experience. You said what are the experience weaknesses. Many pressure students, yeah, experienced also, they are saying my weakness is only talking, yeah, chatting. So that is not weakness. Weakness, you can analyze yourself. What weakness is giving uh, you, yeah, company loss, making the loss. So that you should be tell. When interviews ask this question, it is a genuine way to find out about your flaws and see how you deal with them constructively and how you are decided to improvement. Try not to think of them as a weaknesses. More areas are for improvement. Avoid avoid classes such as I am pre, pre perfectionist. Perfectionist. Use this question to point to strength. For example, use the value and standard of the British Armed Forces, which are admired by the employer by saying something like, I do struggle dealing with, with people who do not share the same value and standards. In my case, those instilled in me by my services in the armed forces turn a negative into the positive. If you can highlight weaknesses and then explain how you are working on improving with examples, the employer will be impressed. Do not pick out a key skill that it mentioned as a requirement of the particular job role as your weaknesses. Why do you want to change the job? This is for experienced guy, is not for freshers. Where do you see yourself in coming 5 years? This can be translated to what are your career goals within this position. That is within interviewers, we want to know. Keep your answer general. Don't be too specific. Ambitions and set goals are important, but if you are too specific, you run the risk of stating goals that are not achievable within the position you are interviewing for. Be truthful but broad enough that it shows you would be good fit for the organization. Trace your interest in long-term career with the company. How would your colleagues describe you? Ask them and prepare accordingly. Have you ever had 360-degree evaluation? Leverage it. What quantities are the interviews hope to identify in the successful candidate? Infer those attributes. What did you like most, least about your last job? This is for again experience. Paint a picture. Paint a picture. Use example of doing relevant work. Those doing the interviewing. How to visualize how you would be fit in and add value. Try to move the focus onto future with the past being used slowly as corroboration. Tell me about a mistake you have made. Be pretty you about that you did wrong and spend a very small proportion of your answer on it, but very clear about how you rectified things, making sure you end on a positive note. You want to leave the impression you learned a significant lesson, ideally one relevant to the role for which you are interviewing. Describe your ideal work environment. It isn't rocket science to out work out what answer and interviewers are looking for in the context of their specific role, but you need to have thought if you are going to articulate them continuously. Example include challenges, collaborative, opportunity for professional development. Describe a time you had difficult within a team. You listed you worked it through, things turned out fine, and you learned for it. Why should you hire you? So this interviewer job is to dare the best person for the role. So this question is particularly important as it can help set you apart from your competitors. This is your chance to truly impress with reasons why they would hire you, why you stand out from the crowd, and a chance to reiterate your strengths. Brainstorm, review the job description, and look at your CV and decide what are the most important qualification, experience, skill, and in which of these area do you really excel. Think about what are your most impressive accomplishment and what make you stand out for other candidates. Keep it in the 1 or 2 minutes range. Along like all the questions, it is important to practice and answer and to come across enthusiastic and confident. You don't want to end up sounding like a robot. So practice so you are confident and comfortable answer the questions of the cup.
So
And
there is a question from Madhubala Bhatia. How to answer a question that what is your expected salary as a fresher in company or organization?
Okay. So it's a very nice question because they don't know what company is giving and what is market rate is going on. So I will giving one example as per India trend in biotech and pharma. In biotech, current MSc microbiology salary now going is not more than 15,000 rupees per month. Okay. And for pharma, not more than 12,000 rupees. This is the average range of microbiology salary. If expected, because management was clearly given the fresher salary is structure to HR. They can answer the question like this: as per company policy and recruitment policy. They should give like this answer. Because company policy is all freshers are same.
Sir, एक question और है sir. Riha Vibhuti Matka, when asked why should we hire you, can we describe whatever the skills and achievements we have written in the CV?
Okay. So why should hire you? That one question, this question, again, this is a good question because hiring person is asking why should hire you? So there is why this question is asking them, because they are checking their confidence. What after hiring, what you are giving to company. First, you say like this: I am having my confidence there, hard work, and I'm always learning attitude. These three answer is fit for there, and I always do for company. Okay. Another, I want to tell you, because we are also growing from bottom to top, how to apply, yeah, how to identify the company and how to submit the CV to company. My suggestion to all students: first, you list out the name of company in India. First, you go like you put into Hyderabad. What are the small company and what are the large company? How you are going to identify the small company and what high company? You go to all Google literatures. There, who is having more than 100 crore turnover, that is all are high big company. If less than 100 crore is very middle company, and less than 50 crore is a small company. So if you want to try for bigger company, you should be know all questions. You should be answer the all questions. At least 80% is a passing criteria for multinational company, whatever they are asking questions. I'm giving one example. If you are going to QC interview as a microbiologist, then you should be know related sterility, related to water testing, related to your staining, bacteria, fungus, then your aseptic techniques. If you read the WHO GMP guideline, they are clearly mentioned there. TRS, multiple TRS is there, USP also there, USP 198. So these guidelines they should read, because like me, I'm a senior, but I don't know basic of microbiology because I forgot last 35 years, 25 years back. I read whatever I read that maybe you don't have. So you should be go for current practices, WHO GMP guideline, current practices. That's what they are saying for microbiologists. Hello.
Yes, sir, you are audible.
So these things they should go for before going to interview. They should read the WHO guideline and related their practices,
That's the practical knowledge.
practical knowledge. If you are going direct theoretically knowledge, nobody asking you theoretical questions. Like only some questions as a microbiologist, they are asking name of scientist and who is going to produce this type of petri dish and why they are calling. Generally, and another most important thing, don't write what you not done in, don't write in your CV what you not done in earlier also, your practical time also. Only naming of equipment is not increasing your CV value. If you really know that equipment details, then you add it. Like microscope, you should be know from their practical problem also and your practices.
So Sunil is
Procedures.
asking that, sir, my English is not at par with the industry standard. Should I stop applying for MNCs and focus on Indian companies?
Okay. There now after COVID, nobody is asking for your languages and all these things. They should go for their own language. At least Hindi is most required. You directly doing only because in all states, their own language science graduation was started.
No,
Like
for
Andhra
MNCs, he
Pradesh.
is asking. Multinational companies, he is asking, sir. Not for
No, he's
India.
asking, but that much English they knows. If they knows in Hindi of giving answer, they should directly say, sir, this is I can give
Explain in
answer
Hindi. In Hindi.
in
Okay.
Hindi. Hindi English, they should openly tell.
Okay, okay.
And one more thing I will say, in CV, what you know is that you write. CV speaks more than you what you want. That 5-6 questions they are asking based on only
Your
your
CV.
CV. Generally, I'm telling you why that 0.2 micron filter is required for sterility. Okay, nobody knows why that 0.2 micron. But here, technical question in BSc and MSc microbiology, our microorganism diameter is less than more than 0.2 micron. So that is this is common answer. But maximum students don't know the 0.2 micron filter is why they are using. They are giving answer like the liquid material is passing, and then is no. Only the answer is 0.2 micron membrane filter is not filtering the bacteria because bacteria size of filter is filter size bacteria size is more than 0.3 micron in liquid stage. Then same thing, what is HEPA filter? Is WHO guideline is writtenly clear what is the HEPA filter, what size of HEPA filter using in production area. Then what is difference between cross contamination and contamination? Contamination means directly growing the bacteria in one product. Cross contamination means earlier product and present product. If those product mixing in and getting result in current batch, that is called as cross contamination.
Cross contamination.
So these simple things is there. They should not be go for high deeply, but only simple thing before going to read all these things. Another thing I will say how to identify. I will suggest to all freshers, first you go to small company because they need freshers.
So
They are
one more
giving
question
small
is
scale.
there. One more question is there, sir. If they ask why Dettol kills 99.9% germs and not other 0.1%, what should be our response? My friend got this question in a recent interview. Sunil is asking.
What question is? Please.
If they ask why Dettol kills 99.9% germs and not other 0.1%, what should our response be? His friend got this question in a recent interview. Sunil is asking, sir.
Okay. Actually, Dettol directly, whatever we are using, they are not killing 100%.
100%. Yes.
Because all disinfectants never kill the 100% bacteria or fungus. If you are giving the time for killing, like I'm during our method validation and this cleaning validation, what we are doing, we are applying Dettol after filtration of 0.2 micron. Okay. Because whatever Dettol we are taking, that may contaminate the growth. That's why we are, before going to apply on injectable area, we are doing the 0.2 micron sterile filtration and then we are applying. If you give 10-minute time after application, if you give 10-minute time, then bacteria killing is there. If you apply and after 5 minutes, if you clean, then maybe 30%, 40% bacteria will be killed. That's why 70% IPA is using as a temporary hand sanitizer. Because if you use 65% IPA, no sanitization is there. If you use 72% also IPA, no sanitization is there. Only 70% because 70% and 30% of water 70% IPA and 30% of water that penetrate the all your membrane and cell wall of bacteria. That's why the effectiveness of disinfectant at 70% is good. Any questions?
Yes, sir. Riha Vibhuti Matha is asking, what to do when they ask us a question related to academics and we don't remember the answer at that moment? How should we cover up, or what should be our response?
If they don't know that, then they should take their biodata in front of them, na?
You should keep a biodata with yourself.
Yes, no problem. Because some years don't know, yeah, percentage of marks don't know. Then you don't say percentage, then say class, second class, first class. That is a vague answer. Sorry, is a not correct accuracy answer, but approximate answer. And if you don't know
Any more
any
questions?
answer, hello? If somebody don't know any answer, they should clearly mention, sir, I don't, I will not answer this question, because you say clearly, sorry, I don't answer.
Any more questions? Someone want to ask questions? You can ask directly also. Sunil is asking, sir, what is life according to you?
Huh?
What is life according to you?
Life.
Life. What is life according to you?
Okay. So my experience, I'm telling now and coming 15 years, pharma and biotechnology and
microbiology,
construction business. These three businesses, बोलते हैं ना, जब तक धरती में आदमी है तब तक ये बिजनेस रहेगा ही, and that is excellent business. Life, if you are saying life, I'm telling you, काम ही भगवान है, ये जो लोग मानते हैं वही लोग लाइफ में सक्सेस होते हैं. अगर आपने काम को ये सोचा कि मैं मैनेजर हूं तो मैं टेबल क्यों क्लीन करूंगा, मैं वाइस प्रेसिडेंट हूं तो ये पोछा क्यों मारूंगा, झाड़ू क्यों मारूंगा, एक ही लाइफ में याद रखना है, मतलब दिस इज आवर स्लोगन, जीवो जीने दो. अगर आप काम करने जा रहे हैं तो काम ही करना, एक तो एक नंबर करना, नहीं तो नहीं करना, बीच में नहीं रहना. किसी ने अगर आके आपको बोला अरे यू क्लीन दैट टेबल, they should come and say yes, wow, क्या क्लीनिंग किया है. Like that everywhere work. If you are everybody appreciate there, then definitely.
Sir, Madhuwala is asking that during interview sometimes nervously we answer wrong, or we become so answer that we don't answer any of few questions in the interview. That makes wrong impression. How to cope with the situations?
Shama ma'am.
Yes, sir.
Before answering the question.
Yes, sir.
You are an admin now.
Yes, sir.
So you remove Sunil.
Okay.
Because he is an idiot fellow, Sunil.
Okay, sir. Okay, sir.
Yes. Yes.
Okay.
Anil, you can start.
Okay. If that nervousness is also there, then after completion of your interview, you can say, sorry sir, whatever I know, that only I give an answer. Whatever I don't know, I try to reach to your expectation, but I feel I am not reached to your expectation. Thanks, but you consider me. I will definitely reach to your expectation by showing our workplace.
Hello.
Hello.
Hello, sir. I have one question.
Hmm.
I am searching a job from 2 years.
Hmm.
No any vacancy in any company. I apply so many company, but no reply.
Okay.
So how they are apply when they reply me?
Okay. Where
Because I
now
have so
where
many times apply in pharma company, food industry.
no
So
where
please,
you are staying, where you apply, and how you apply by mail
Aurangabad,
only.
Aurangabad, Aurangabad
Aurangabad.
city
You
of
are staying in Aurangabad.
North.
Okay.
Yes, sir.
Okay.
Mm.
So here, what it is a very good question. I will say, if you are applying and if you are not getting reply to you, you first check your biodata, what you written in your biodata, and which industry. Same biodata cannot work for all industries. Okay, if you are microbiologist, you check that what vacancy is there,
Mm.
or and Aurangabad is not pharmaceutical industries background. Aurangabad is only API, food industries.
Mm.
Okay, if you want to go for food industries and background, Gujarat, now Nashik, that Nagpur area, Pune,
Mm.
Mumbai, Nashik, Hyderabad. That's why I want to say all students. First, you choice what collect the data from Google.
Mm.
Okay, that Google give you
Okay.
all data, which industry is where, what industry is there, what industry is manufacturing. If you go with that,
But I
okay,
searching, sir. So many times searching. I apply also Pune, Nashik, all the state, and I so many times my biodata are repair and resume is re-update from site LinkedIn, Naukri.com, all of these
okay,
sites, but
okay.
nothing
I will one
replies
more.
anything.
I will give one suggestion. You go for system statistic SAS. Maybe you know regarding this. That is a 6 months courses is there now. That is used for IT industry also. If you are postgraduate, a graduate in science, if you go with
Mm.
SAS, system
Yes, yes.
analytical, uh, uh, sorry, statistic analytical system. That is the courses. Different courses is there. 6 month
Oh.
courses is there. Based
Mm.
on your technical things, you select that your what you want, and that courses is required the
Mm.
analyzing the data, because all company
Mm.
required generate the data,
Mm.
acknowledge the data. Even small hospital also they started data analysis.
Mm.
That's why now statistic people is a very big importance.
Yeah.
How I
Again,
can read this? Is suppose.
again, you
Uh,
see, I want to answer her question.
Please, sir.
Yes,
If
sir.
you are applying continuously,
Mm.
but not getting response from any industry,
Mm.
but thousands of students are getting job in the industry. So you ask yourself, why not you are getting? I will tell you the answer.
Mm.
As Dr. Anil has told, you yourself improve more, update more. You make fit for job. How? By doing certain certificate courses. He has told statistic is a very important. Bioinformatics course also. In food
Yeah.
microbiology, quality control course. So like that, various courses which industries are requiring, you should increase your CV. And one more thing also is important. If your senior is already in the job, you can contact the senior, he will introduce in the company. But if you don't have relation with the senior, then improve your biodata. That is the only way.
Sir, I
So
will
you
give.
think why you are not getting, and that's why you improve yourself. That is the answer. Okay.
Sir, I can, if you share his biodata, her biodata, she
Correct.
is ready to work in this near to Amravati. Ice cream
Mm.
factory is there.
Mm.
Very good, very good.
Okay, and they are start. My friend was started that ice cream factory. They want a lady like who is managing the all ladies in packing line and process line.
Okay, then it is very good. So
He
you
will
can
give the
contact with Dr. Anil.
accommodation free and 15,000 rupees salary.
That's very good.
Okay.
If you want to
Okay.
really grow their career, so they should go like this also, because what you are suggested, दुनिया में जब तक आपकी कनेक्टिविटी नहीं है, रिलेशनशिप नहीं है,
Correct,
तो
correct.
नेवर ग्रो.
You will never grow.
शायद वही
मैंने जो
problem है।
बताया ये गुरु मंत्र है, समझ लो.
Yes.
One more question is there, sir, from Pramod Kumar.
Yeah.
Genetic engineering are important. Which industry more important, biotechnology or pharmacist?
Biotechnology, genetic engineering. That is now new strain is coming. Trend is coming for biosimilars.
Yes.
So all companies started biosimilars in India, so that DNA, RNA technology, genetic technology is required there. But this is R&D, again R&D trend. मैं आप सभी लोगों को एक रिक्वेस्ट करता हूँ, मैं बहुत इंटेलिजेंट नहीं हूँ, ओनली नॉर्मल मार्क्स मिलने वाला हूँ. जॉब कैसे मिलना, कैसे करना, मैं एक एग्जांपल बताता हूँ.
Yes, sir.
मेरा करियर मैंने 1994 में स्टार्ट किया. मैं मेरे विलेज से बॉम्बे चले गए. आई एम नॉट टेलिंग, मतलब ये चाय वाले का स्टोरी नहीं है. रियल जॉब कैसे लेना, आपके पास क्या होना चाहिए. जब मैं 94 में मेरा ग्रेजुएशन कंप्लीट हुआ, तो मैं बॉम्बे चला गया. मेरे लाइफ में बॉम्बे कभी गया नहीं था. तो मैं थाना स्टेशन पे जब उतरा, तो उधर एक इंटरव्यू दे के आया. तभी पोस्ट बॉक्स से इंटरव्यू चलती थी. पोस्ट बॉक्स में नंबर आता था, उसके ऊपर इंटरव्यू जाना. तो मैं इंडस्ट्रियल सॉल्वेंट एंड केमिकल का एक नवा काल में एडवर्टाइज देखा था. तो उसी एडवर्टाइज पे आई वेंट टू इंडस्ट्रियल सॉल्वेंट एंड केमिकल्स near to Thana Station Mental Hospital. That company is there. I visited that company. I was given interview, but I don't know Hindi and English. Then that manager, works manager is highly South Indian, and he was asked me question in English. I was given really Marathi answer, whatever I know. Then he asked, you don't have experience, why you are, we are not considering you, sir. कोई तो किसी ने मेरे को जॉब देगा तब मैं एक्सपीरियंस दूंगा. मेरे पास एक एक्सपीरियंस है खेती करने का, एक्सपीरियंस है मतलब फार्मिंग करने का, या गाय भैंस के संभालने का एक्सपीरियंस है, दूध बेचने का एक्सपीरियंस है. but I don't have other experience. जब तक कोई देगा नहीं, then immediately he will appreciate and he give me job. Same day he told me, tomorrow you join there. What purpose he joined? Because I clearly mentioned, in my mind, whatever coming, I was saying, I don't have experience, I accepted. But what experience I have, that I told him. So he want a hard work person. That's why he was giving job to me. Again, after 1 month, 2 months, I asked him, so why you are giving job me? बोले कि आपका आंसर इतना अच्छा था कि किसी ने काम दिया तो मैं एक्सपीरियंस आएगा. तो ये चीज क्या होती है कि आपका कॉन्फिडेंस बोलता है, आपको चाहिए क्या, आपका गोल क्या है, आपको क्या चलना चाहिए. कभी कभी क्या होता है कि आपको गेट पे जाते, अगर अभी जैसा ये जो लड़की आपको जो क्वेश्चन पूछ रही थी स्टूडेंट, उसने अगर औरंगाबाद छोड़ के पुणे में जाके अगर इंडस्ट्री घूमी, हर गेट पे जाके बायोडाटा दिया, सिक्योरिटी को पूछा बाबा वर्क मैनेजर कौन है, इधर प्रोडक्शन मैनेजर कौन है, किसी में कॉन्टेक्ट नंबर लिया, जैसा सर ने बोला कि आपके सीनियर्स का कॉन्टेक्ट छोड़ दो. अगर आपके सीनियर्स नहीं है कुछ कारण से, या एज ए बीइंग लेडीज मेनी फ्यू रिलेशन इज देयर, तो अगर नहीं है तो जाओ गेट पे, सिक्योरिटी को पूछो. उधर अभी आजकल बोर्ड भी लगे रहते हैं, वर्क्स मैनेजर सेफ्टी ऑफिसर का नंबर रहता है, यू टेक दैट नंबर, कॉल देम.
हेलो सर, मैंने एक फोस्टर जो कंपनी है वहां पे अप्रेंटिसशिप की थी 2 साल तक, बट वहां पे कोई ऑन रोल जॉब नहीं दिया उन्होंने हमें, और इसलिए मैंने वो जॉब छोड़ दिया और मैं पुणे शिफ्ट हो गई. उसके बाद मैं फिर से औरंगाबाद आ गई, मैं फिर से जॉब सर्च करने लगी. बट प्रॉब्लम ये हुआ कि ऑन रोल जॉब नहीं था मुझे, इसलिए, और मैं ऑन रोल जॉब ढूंढ रही थी बहुत दिनों से.
नहीं, मैं आपको इसके लिए बोल रहा हूँ, एज ए माइक्रोबायोलॉजिस्ट ऑन रोल. देखो दुनिया में कोई आज के तारीख में after coming this Modi government, no this on job, on roll and out roll. Only PF difference is there. Nothing is changed. आप कई पैसा काट के गवर्नमेंट में रखते और छोड़ने के बाद आपको देते. On roll and off roll nothing is changed. ओनली 2 टाइम का खाने में कुछ 2 रुपए कूपन का सिस्टम में प्रॉब्लम है. आप पहला जॉब को जो है उसको कंटिन्यू करो. अगर आपको ऐसा लगता है जॉब नहीं मिल रहा है, आई विल गिव गुड एग्जांपल. यू कम हैदराबाद, पर मंथ 5,000 रुपीज पे हियर फॉर 2 टाइम खाना, लंच एंड रहना. अच्छा, इधर हॉस्टल से लेडीज हॉस्टल, और हर दिन 3,000 कंपनी है हैदराबाद में. फार्मा, बायोटेक, टैबलेट, कैप्सूल. हर गेट पे जाओ कंपनी में. 3,000 कंपनी आपको घूमने के लिए कितना दिन लगेगा? 1 महीना पकड़ो, डेली 10 भी घुमा तो उसमें से 3,000 में से एक तो पक्का आपको देगा, उसको उसी दिन जरूरत भी हो सकती है. पुणे और देखो, आप 2 सिटी सिलेक्ट किया, 2 में अगर आप आपका खुद का कॉन्फिडेंस अगर लूज किया कि जॉब ही नहीं है, you go to you start your own job also. Own also start, you go for take the tuitions.
Yes.
या आप एज ए माइक्रोबायोलॉजिस्ट अगर ग्राम पंचायत का सरपंच को मिलेगी और लेडीज को अवेयरनेस करेगी फॉर देयर प्रैक्टिसेस, डिसीज प्रैक्टिसेस कंट्रोल, तो उनमें अगर आप वो भी करेगी तो आपको कोई भी अच्छा इनकम होगा, आपका नाम होगा. You are coming on socialism. You choose that also. NGO is there. Multiple NGOs required, like you people expert in microbiology.
One more
NGO
question.
is there.
One more question is there. Nitin Pahade.
Now, ma'am, you take last question now.
Okay, sir. Actually, for freshers, it is little bit hard to get a job in any industry as compared to the experienced person. No, Nitin, it is not like that. I am answering that, but afterwards, sir and Deshmukh sir will answer that no, it is not like that. That freshers only, freshers doesn't get the job, but experienced persons are only required. There are many industries which allows the freshers to come in because they are actually very energetic. But your biodata should be up to mark. You should have that internship. You should have that certificate courses. You should have other courses. Then the year required pro forma of teaching. Means जो हम तुम्हारे कोर्स में है उसके अलावा अगर थोड़ा सा भी कुछ होगा, थोड़ा सा भी कुछ हो, तो you will be absorbed. That is my view. Sir, Arvind sir and Anil sir.
100% correct, madam. Only theoretically knowledge now industry is not going to accept. They
Yes,
want
sir.
some practical knowledge like small courses was. If you go for SAS, definitely companies are taking you.
You just don't stick to your course that we are completing our PG and we will get the job. No, you have to do some of the other type of the skill-based work, some value-added courses, some certificate courses, and spend money. That is your investment at this time, which will pay you later on.
Yes, sir.
One more thing, ma'am.
Yes, sir.
In young generation, there is a belief
Yes.
that I should get initially 20,000 rupees per month.
Sir, you are saying less amount. They are thinking about 50,000. That
Yes.
package. Yes, yearly package.
In the beginning
yes,
only.
Yes, in the beginning
So,
only.
I am requesting the student.
I started from 800, sir. I started from 800.
Yes, you start your job. Whatsoever the salary is not
Whatsoever the salary. Yes, sir. Yes, sir. Because then only you will gain the experience.
Correct. So in the first job, experience is important. Salary is not important.
Not important. Yes.
So that should be, many things are there. But today's Anil's lecture is very enlightening. As students might have enjoyed, and again, I'm circulating this YouTube in 20,000 students.
Thank you, sir. Thank you.
Okay,
So student will enjoy it and will contact you as you have given the telephone number and your mail ID.
Yes, definitely, sir. I will. Last year you told me during COVID.
Correct.
You have given my number. So almost 230 biodata I received. Out of that, 130 people I gave job. In my company, only 80 student is there.
There are 2-3 questions in the chat box, but I request them that you write with the mail to Dr. Anil. Now
Yes.
we'll conclude now. So Shama ma'am, you can proceed.
Yes, sir. Yes, sir. The rest of the questions, students, please ask to sir at his mail ID, or you can contact him personally also. And thank you very much, sir, Anil Yadav sir, for being with us and giving us a very nice presentation about how to prepare. The student should how to prepare for the interview, as many of the freshers, they don't know what the questions will be asked. So as an entrepreneur, you have also told us that these are the questions which when you are going for a microbiologist place, these are the questions they will be asking. So get your practical knowledge very well hand, and you should try to tell about your abilities, about your work, about your experiences which you have had during your studies, and what you can do. You have to present yourself, your good things, and which sir has told very well, in a very well mannered, with all the PPTs and slides. You should have know all the answers also. And your CV, you should try to maintain your CV in a good condition. Next, I will ask Arvind sir to have a seminar or a webinar to how to write your CV, how to improve your CV, how to make your CV more glorified, because nowadays जो दिखता है वो बिकता है, और जो दिखता है वही चीजें जो है वो आपको आगे लेके आएंगी. हो सकता है आप बोलने में थोड़ा झिझकते हो, तो जो आपकी कमी है वो आपको मालूम है, तो बेटर आप उस कमी को दूर करने के लिए अपने टीचर्स से बात करिए, वो आपको कुछ पॉइंट्स देंगे. या हियर देयर इज अ गुड प्लेटफॉर्म. You can directly ask to Deshmukh sir also, and the entrepreneurs which we are contacting every Sunday. We have started this series. And so thank you very much, Anil sir, for joining with us in a very short time of time given to you. Thank you very much, sir.
Thank you.
Thank you to all the students also, Deshmukh sir, for his blessings for
Shama
conducting
ma'am,
this program. Yes, sir.
you can stop this video
Okay,
because
sir.
you are admin now.
Okay, sir. Okay, sir. Thank you very much. Thank you to all.
Thank you, Deshmukh sir. As my guru, he was given opportunity to me connect this platform and express my experience. I will try to do more good PPT for production, for QC, for QA, how get the job, and I will share through Deshmukh sir. Some of companies routinely they are required some
Microbiologist.
So I will share that day-to-day interviews dates. So sir,
Yes,
to everybody.
Dr. Anil, you deserve for it. That's why you are asked to present a PPT, and many students will get benefit. Thank you.
Thank you very much, sir. Thank
आपने
you.
हमको गुरु विद्या दी है, हम आपकी सब ज्ञान दुनिया को दे रहे हैं, बस nothing.
Yes, yes, yes.
Thank you to all. Thank you to all.
Thanks once again.